Hello, everyone, and welcome to today's webinar, Using MERFISH to Decode Spatially Regulated Disease Drivers in Human Heart and Kidney Disease. I am Kaylee Walk of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Vizgen. To learn more, visit vizgen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speakers, Dr. Christoph Kuppe, Professor, Principal Investigator, Laboratory of Quantitative Cell Dynamics and Translational Systems, Biology, University Hospital, RWTH Aachen, and Carol Shadid, Field Application Scientist at Vizgen. And also joining us for Q&A today is Joe Maffey, Field Application Scientist at Vizgen. For a complete biography on our speakers, please click on the Presenter tab. I'll now pass it over to Carol to get us started.
Hello everyone, thank you for joining us for today's webinar. My name is Carol. I am a Field Application Scientist at Vizgen. I'm based in the UK and I will be giving you a few minutes introduction just so you know who we are and what we do before leaving the floor to Dr. Christoph Kuppe, who will present his work on MERScope. And just for your context, Vizgen is a company that's headquartered in Cambridge, Massachusetts, in the US. It was founded in 2019 as a spin-off from Harvard University, and it originated within the lab of Dr. Xiaowei Zhuang, who, with her team, developed MERFISH. And there's been quite some growth since 2019. We are over 200 employees as of September 2023. And we are a spatial genomics company. So what does this mean in terms of our mission? It means that we are dedicated to pioneer the next generation of what's going to be happening in the field of spatial genomics. So that means working on new tissues, kind of uncharted territories, new processes, new protocols, really trying to push the limits of what we can do currently to get better data and deeper insight in tissues that are both healthy and diseased. But why spatial? So if I take this really simple example, you can see all these different tiles individually are very rich in information. However, it's really hard to make sense of whatever's happening in the screen because they're all scrambled up. So information without its spatial context is information, but it doesn't really offer the insight it could, as we can see here. So if we move from this really simple example and kind of try to translate it to the genomics field, put it back into some historical perspective, we are kind of all stemming from some work in bulk sequencing, which was really brought a lot into understanding biological systems as defined by the expression of many genes. And it was really more about comparing gene expression between one tissue and another, or between the same tissue that's in different states. It was going through extraction, high throughput sequencing. It's still a huge part of the work that we do in the genomics field. And then there's kind of started to be moving towards single cell based analysis. So it really helps us understand biological system as being compartmentalized into individual cells and compare gene expression between two different cells. So based on this, it's quite straightforward to understand the extra layer of information that single cell spatial genomics bring. We're still working in the single cell mindset, but we're adding in situ detection, so we really understand how biological systems are spatially organized. And not even just that, we also, with our technology, we can see within a cell where the transcripts are, and we can fully quantify this. So it's really adding an extra layer of spatial information. So just to help you understand what kind of data you can get with one MERScope run, let's try and compare it to this map of New York City. So let's say that one transcriptomic run with MERScope is this 100 by 100 kilometer map of New York City. So you can really kind of see where you are. You don't have any details, but you know you're in New York. You can see Newark. You've got 1 meter resolution. So this is what we would call kind of like a whole tissue view, kind of overview, what organ you're working on, what it looks like. Then you would be able to zoom in to what we would call a whole field of view, kind of like type of zooming in. So here you can start seeing some details. You can see the rivers, you can see a park. So this would be equivalent to something like 200 by 200 micrometers on the MERScope run. But then it doesn't stop there. You could also zoom into really deep into your tissue. So for example, here I zoomed at the corner of 5th Avenue and 58th Street. So I didn't make this slide, but I actually went and see what was there, and it's actually an Apple store. So obviously you won't have that in your MERScope data, but this is really where you could see transcripts within your cell, nuclear structures, with a resolution of 100 nanometers this time. So this is all of the data that comes per run. And this slide kind of summarizes what I just said, just so you can understand a bit more. This is a mouse brain tissue that was imaged with MERScope, and we go progressively from whole tissue to wide field of view to subcellular, where each of the dots corresponds to a transcript. So if we kind of put it into more perspective, MERScope, which is our instrument, is a proprietary platform for MERFISH. So there's loads of people doing MERFISH, there's hundreds of MERFISH papers. Vizgen's aim with MERScope was to make MERFISH accessible to researchers. So we're working on a solution that includes everything that's needed to quantify and localize transcripts in any tissues. So that includes obviously the instrument itself, the analysis computers, but also everything from upstream, including panel design, pre-experiment support, kind of talking you through how to work with your tissues, all the way to kind of downstream. So we have an array of interactive visualization tools that let you look at and analyze your tissue and really make sense of this wealth of information that we've seen comes from one MERScope run. So really trying to connect MERScope, MERFISH, Vizgen. We are working to bring you MERFISH technology in a really streamlined fashion. So that's just it for me. I hope this gives you a bit of insight about what we do, and I'm happy to let Dr. Kuppe talk about his work. Thank you very much, and talk to you soon.
Great. Well, thank you so much, Carol. Now, Christoph, we'll pass it over to you.
Thank you very much for the very kind introduction. I'm very happy to talk to you today about using MERFISH to decode spatially regulated disease drivers in human heart and kidney disease. So why do we need this very high resolution spatial transcriptomic approaches? So with spatial technologies advancing in their capabilities, we are able to ask further questions. And some of those I've listed here from a very good review article published in Nature Biotech last year from Evan Markosko's group. So these very high resolution technologies like MERFISH allow us really to map not only the cell state location, but actually also give us a very good measurement about the cell composition within any given tissue. However, since we don't disrupt the tissue, we can use this data, of course, also to study cell-cell interaction. And this can be anything like decoding spatial dependencies of cell states, or analyzing distinct neighborhoods, which are important, for example, to guarantee homeostasis of a tissue, like, for example, in the lung, in the kidney, or in the intestine. But we, of course, can also go a step further and study not only cell-cell interaction, but actually molecular interactions, because we have the subcellular information about the transcripts, and we can basically use this as a model to look at receptor ligand expression and also the dynamics of these processes within a given cell. For example, very excitingly, for a tumor microenvironment where you might have a specially engineered cell therapy to target the cancer cells. So this technology then allows you to really dive deep into the situation and analyze, identify potential biomarkers that allow you to, target this even more specifically. I think it's important to generally divide these high-resolution spatial transcriptomic approaches into sequencing-based or imaging-based approaches. Sequencing-based approaches in general destruct the tissue, and a DNA library with spatial barcode is generated and sequenced on a high-throughput sequencer. And since these spatial barcodes have unique identities, one can then later reconstruct and get two matrices, a gene expression matrix and a spatial information matrix. Typically, the sequencing-based technologies are not at a molecular resolution. Some of them can be at subcellular resolution, but the only molecular resolution technologies, all of those are imaging based, including MERFISH. So here you would really get within a cell the subcellular resolution of individual transcripts or potentially even other biomolecules that you target. So MERFISH is an imaging-based technology. And MERFISH was developed by Professor Xiaowei Cheng, a professor at Harvard, and in her and her team's seminal study published in Science, they introduced the concept of MERFISH. So MERFISH stands for Multiplexed Error-Robust Fish. And this multiplexing and error robustness, these are two features which are really unique to MERFISH in this combination. So there are lots of multiplexing fish technologies out there. However, if you look down here in panel B, C, and D, you can see the black lines. These are theoretical considerations when you barcode and encode a code book into the transcripts that you want to detect and really scale this up. You can then see that the error or misidentification rate of those transcripts actually increases tremendously. So you need to have a mechanism, a design of the code book to allow you to really confidently and with a very low error rate detect those transcripts at a subcellular resolution. And this was really the innovative part in the recipe to get all of this working. I would say, among lots of other things, of course. But then how this works is that you, in MERFISH, images are generated, for example, from cells and cell cultures or entire tissues. And then you, in several imaging rounds, a spatial barcode is read out from the cells. And indeed, this is a binary barcode. So you either detect a signal, then it's a 1, or you don't detect a signal, then it's a 0. And this is performed over n imaging rounds, giving you this bit barcode, and each barcode is unique to its transcript, and you have to define this up front. In their study, they also show how the raw data basically looks like. So here you see an example of a hybridization round. You see all of those spots here, which are positive signals. And then a bleaching is performed of the signal in a flow cell, basically on a microscope. And then you see a hybridization round 2. And then this is performed several times. This also, of course, increases then the imaging time. The further hybridizations you do, you can see here 16 rounds, like a 16-bit barcode. And then you can reconstruct the spatial positions from decoding these images. And this process is highly reproducible and very well correlated to bulk transcriptome data, also in the lower ranged, lowly expressed range transcripts. And I want to go a little bit more into these barcodes design because I think it's essential for MERFISH. So up front, if you design a panel for the MERFISH experiment, you have to, each RNA target has to have a unique binary barcode. this unique binary barcode is made up of zeros and one, and then is assigned to an individual RNA target. So this here, for example, are up to 140 targets, and each of the targets would have a different binary barcode, right? And the bits, so the numbers within these barcodes are always going to be the same for one codebook, basically. So for example, for a 140 gene panel, those would be 16, 17, 18 bits. And then what happens is that we hybridize this panel to the tissue sample, perform a gel embedding, a tissue clearing, and then basically everything runs nicely on its own on the MERScope provided by Vizgen. And these signals or no signals are actually recovered from the polyacrylamide gel. And per target, these encoding probes bind. There's not only one probe binding to the RNA target, but several probes, guaranteeing that the signal is actually strong enough, because this has to be, these signals are not amplified. So you need several binding areas. And these can be, depending also on the abundance of the transcripts, 1 to 50 probes. And then at the moment, one can target 140, 300, or 500 different transcripts with this technology. Now, on the instrument, each bit position is actually read out individually. So let's say in bit position 1, we get the images, and target RNA, the RNA target 3 generates a signal, and the target 6, whereas the others don't generate a signal. So this is on this side here how this would look like. A 3 and the 6 would generate a signal. This will be recorded and stored. And after this imaging round, which goes across, of course, the entire tissue section, afterwards, all the signals are then photo-bleached. And then we would go to the next round, or the instrument does, of course, this nicely automatically. And then another, either signals or no signals are recorded, photo bleaching takes place, and all of this is done automatically on the machine. So then, after all of those rounds of hybridization and photo bleaching, one actually gets this this barcode, which is analyzed automatically on an analysis computer. And you can see here that these fluorescent signals, which showed up in position 1, 2, 6, and 9. We know this from our codebook. They are actually encoded by the target 6, and anywhere on the entire tissue section where we can identify this codebook, we know that the target 6 was RNA species was present. Another important feature is the error robustness, and for this, in the codebook, a certain design is included, guaranteeing that errors in the sequencing can actually be corrected. So between two different words, for example, so this is RNA transcript 1, this is 2, there's at least, with a Hamming distance of 4, there are at least 4 different signals, basically, of zeros and 1. And all of them actually would have to be converted so that we read out a different word than we are supposed to. So meaning that if there's a single error, for example, we can still correct it into the correct code word, right? And I think this is very unique. However, if there are like really double errors occurring, then of course we at least know that that there was an error in the detection, and we can discard this. And this is not possible with other in situ sequencing technologies unique to MERFISH. And then what this allows us to do is to look at the subcellular distributions of 100 to thousands of RNA species, of course, not only in cell culture, but really across tissue sections. And I'll show you this in a minute. This data then allows you to look for, basically expression of transcripts and also expression noise. Right, we can look at expression covariation within our data. We can localize transcripts in different subcellular domains of cells and study their spatial distribution. And why is this important? So I'm actually a nephrologist, so I'm seeing patients with kidney disease, and we study we perform the diagnosis based on histological phenotypes. And this is an example here that you can see. Here's this human tissue section stained with trichrome. So everything in blue is fibrosis, and you can see this is a heavily diseased tissue. However, what you also see is that some of the tubuli, these filtering units in the kidney, are actually functioning, whereas others are maladaptive to the whatever triggered the kidney disease. And this spatial correlation, of course, here you can see that these processes can happen in the tissue in a very complex manner. And to really be able to understand what's going on here in all of these different positions, one needs really a spatial single cell resolution. And what we've in the kidney previously asked, and this was not known, what are the cellular drivers of the fibrosis in the tissue. And for this, we used, as this is done for lots of other biological questions, use single-cell RNA sequencing with a high throughput assay to really get molecular information about the different cells in the human kidney in health and disease. And this allowed us to profile not only endothelial cells, different epithelial cells and mesenchymal cells, but also the mesenchymal cells, so the cells which include the myofibroblasts, which produce the scar tissue. And in the end, we were able to find different cell states of fibroblasts and myofibroblasts. But of course, just from single-cell RNA data, it's very difficult to locate those different cell states and also maybe look at downstream targets of their transcription factors that are active in these states, just based on single-cell data where the tissue is basically destructed. And one of the also important questions, of course, is if we have these different states of myofibroblasts, fibroblasts, and pericytes, to which cells, different, there are immune cells, other cells in the tissue. The kidney is one of the most complex organs with over 50 cell types. Where is crosstalk happening? And of course, we can see here, based on this cell-cell communication analysis, that immune cells, not surprisingly, pop up. But you can also see here arteriolar endothelial cells. And of course, this is something that shows up because spatial information of those different cell types is actually missing, not allowing us really to go further in depth and study the dynamics of these cell-cell communication events. But what we would like actually is this sort of situation where we have the single cell information of each individual cells and its cell state and can map it back into the tissue. And this is here a human kidney tissue section where we've just with some marker genes highlighted the capability of this technology to really map out all of those cell states in one tissue. And here's another example. These are podocytes. So these are the cells of the filtering unit, and you can clearly see how nicely these cells actually map to the tissue, allowing you now in the data to compare actually the expression profile for any given glomerulus to another in the tissue. But how best to design these panels, which I talked about? This is actually not a trivial task, but for us, what's interesting, or what we aim to, is really to generate a panel which could map out any sort of cell state or cell type that has been described in single-cell hard data, human single-cell hard data sets. So we integrated the publicly available data set and then used an end-to-end pipeline developed by Louis Kümmerle, Malte Lücken, and Fabian from Fabian Theis lab in Munich, called SPA-PROS. So here you can actually use the single-cell RNA-seq reference and select a probe set to allow you then later to integrate integrate the single cell RNA sequencing data with your spatial transcriptomics data, making sure that you included in the probe set those target genes that allow you to perfectly map and integrate the different cells. However, one would maybe guess that using this approach, you would need not only 100, but several thousands of genes to map out for, let's say, a 300 million single-cell heart data set. However, that's not true. So if we go through here, for example, different fibroblast states, you can see here that if we optimize this using SPA-PROS, this actually works quite nice, giving us those genes that discriminate all of those different fibroblast states. And overall, we just ended up with under 300 genes, transcripts that we needed to include to represent the manifold of those, of all of those cell types actually in this data set. One of the capabilities of MERFISH is actually that it allows us to include FFPE tissue. So FFPE stands for formalin fixed, paraffin embedded, and this is actually a tissue conservation method used by pathologists all around the world. And the exciting thing is, of course, that if you have a biobank and those tissues don't need to be in a -80 or, you know, they just are usually kept at room temperature in an air-conditioned environment, hopefully. We know, of course, that in those tissues, the RNA can be degraded, and this degradation can be sample and region specific. But I will show you now examples of tissues where we actually were quite successful in using MERFISH, which I find really exciting. So this is one of the example of the human kidney FFPE MERFISH that we've done. So here you can see clearly here a glomerulus, different proximal tubular cells, and thick ascending limbs, which are here in green. And you can also see that we are able to capture molecular information from larger blood vessels. And you can see here that the lumen is actually quite small. So this is already a blood vessel transformed by hypertension. This is another view on this data set. So you have here the glomeruli. You can even see that we can get information of the parietal epithelial cells. So these are here the filtering unit where the urine is produced and actually collected here in this space, which is called Bowman space. And lining this Bowman space are cells which are really thin, and we can even with this technology get the information about those cells because they're usually included in glomerular disease situations. Here you have another view, of course, then a next step to look at the single cell information is to perform segmentation, and I'll show you later also a little bit more about that. But this is basically the output from the pipeline, the Vizgen pipeline, already performed and processed on the MERScope after it's run. And here you can also see the subcellular resolution. So you can here, for example, see the organization of some of those transcripts here in this tubular segment. You can clearly see that some of them are clearly in the periphery of the cells, whereas other are distributed all across the cell bodies. I think this is quite exciting and offers a window of opportunity for analysis types that were not possible before. You see another view, I think, on the different cell types we are able to decode in human FFPE tissue sections. Even this here is a tubule. And so here in this area, there's usually urine flowing, right? And there's a single cell here and there. Those are cells that have actually been, how do you say that? They are lost into the urine, right? A kidney loses cells into the urine, and even from those cells here, we can get some information, which also here is another one, which I find really exciting. We previously also used spatial transcriptomics in my postdoc time with Raphael Kramer's group in Aachen to decode the spatial drivers of human myocardial infarction using Vizgen from 10x Genomics. As you might know, this is a spatial DNA barcoding, so NGS sequencing spatial method, however, with a 55 micrometer resolution. Back then, I chose the human heart because per area, so per 55 micrometers, there are not as many nuclei captured by this technology, so, compared to the kidney. The kidney is a lot denser in terms of nuclei per spot. And however, of course, was still very exciting for us was to generate these spatial maps and identify cellular niches. And with this technology, we were able to perform this on tissues from control, but which are healthy heart samples from non-transplanted situations, border zones, ischemic zones, and fibrotic zones. So data from even very heavily diseased tissues, including then the deconvolution using cell-to-location of different cell types, different pathways, and also by including a multiomics approach, the transcription factor activity within the tissue. However, as I mentioned, we don't have the single cell resolution at this point. Now, similar to the human kidney that I've shown you, we are now able to get FFPE information with single cell resolution of human heart in FFPE. So this is what you see here. This is actually a transmural section, like a cross section of a human heart. Here you see actually the epicardial layer. These are fat cells, and these are the endocardial. This is the endocardial layer. So this is from within inside the human heart. You can here see what a pathologist would call an endocardial fibrosis. So it's an accumulation of some fibroblasts in this area. This is a very diseased heart, so the myocardium at this area is quite thin. These here are the myocytes, so the muscle which contract, but you can also see here different vascular structures. If we move further in, you can really see like individual fibroblasts, larger blood vessels. These here are the adipocytes, where you, of course, see black holes because this is the fat that has been stored in those cells, and only in the periphery you see basically the transcripts of an individual cell. So, and this is even possible with FFPE tissue, which I find really exciting. If you zoom in further, you can even see that for some cells, like the fibroblasts, and I'm sorry for this rather lowish resolution image here, but you can actually appreciate that some of those fibroblast arms, you can actually even decipher here using this technology. You can already also see, so for example, this here is one cardiomyocyte, and you can see the transcript variants of nuclear transcript, perinuclear transcript, and transcripts which are further more in the periphery. And I think these are also this you can see here and in other areas of the tissues. And I think this is really the exciting features of this technology. So maybe using this video, I would like to further bring this resolution to your attention. So here we are just focusing on some immune cells, T cells. And if we move in further, we can actually, this is a combination of marker genes, just a very few ones, actually zoom into this tissue and get this information of where these T cells are actually localized. This brings me to my next point, which is actually the segmentation of the data. And here, giving credit to our last year's Space Hack project, which we participated in, where we try to really answer the question, which of those several cell segmentation approaches are best for our human cardiac tissue. You see the results here below. To make a long story short, we were not happy with, we didn't find a perfect solution, but that was also not what we expected. From our impression from this, the CellPose2 model actually worked quite nicely, as you can see here, but other methods actually also spot-based segmentation methods, which don't take any prior into account, were sort of problematic. What I think is really important to optimize cell segmentation, especially in tissue situations, as complex as the human heart are additional protein stains. And this is where MERSCO protein detection, which is a commercial add-on you can buy, not only for the cell borders, but also for additional protein targets. And it works in a way that you establish with any primary antibody a staining. You just have to make sure that this primary antibody is carrier-free, so you don't want to bring in any RNases into your MERFISH workflow, and then you use a secondary antibody and imager construct provided in a kit to detect the location of these primary antibodies. And I'll show you some examples of those stainings in a moment. So here you can see the different hard MERFISH examples of the border stainings. You can see certainly that these two stainings here give us a nice representation of the cardiomyocytes. This staining here is in our hands for this tissue type, not as informative, but this cell border staining kit developed by Vizgen is actually targeting lots of tissue situations, so not only the human heart, but also tissues like intestine or spleen. We haven't tested all of them, and I guess, depending on the tissue type, different stainings will be more informative than others. I think important for a muscle tissue is to keep in mind that some of these myocytes may or may not have actually a nuclei signal included. And this is, I think, important when just relying on DAPI as a prior, because in the human heart, for example, if you use DAPI, and only about 20% of all myocytes will have in their cross-section the DAPI signal. So you would lose lots of data if you don't include an additional border staining. And also, different border staining can give you different information. So those border stainings can be, of course, the cell-cell contacts, and some of these cell-cell contacts are unique, right? So for the human heart and the myocytes, they are myocyte cell contacts, but they are also myocyte non-myocyte contacts, and those are both actually captured with these cell border stainings. Here I would like to share with you a staining we established for the human heart in FFPE using vimenting and a staining for the intercalated disc of the human heart. This is again a cross-section where you here see the epicardial and here the endocardial area. If we move in, we can see that the vimentin nicely also stains these different fibroblast populations and some endothelial cells. This is what you see here. And if we zoom in closer, you can see that using this antibody kit, we can nicely actually decode those these cell-cell junctions between cardiomyocytes. Actually, these are gap junctions. And of course, I think this is data one can, of course, use for downstream analysis, also for segmentation purposes like the vimenting staining. So with this, I would like to summarize a little bit of the data that I've presented to you. So the genome scale imaging approaches allow us now to perform single-cell omics analysis of RNA, protein. I haven't shown you DNA or epigenomics, but that's also possible with MERFISH in a spatially resolved manner in intact cells, and I think most importantly, tissues. These advances allow gene expression profiling of individual cells and an in situ detection and spatial mapping of different cell states in complex tissues. The FFPE material in combination with MERFISH, I think, has a high translational potential to uncover novel disease mechanisms, but also biomarkers and potentially also novel drug targets. We think that the high spatial resolution of a MERFISH also will allow further to determine the spatial organization of the genome, different proteins at high resolution, and definitely already of the transcriptome with a subcellular resolution. So at this stage, I would like to thank you. I particularly want to thank my lab, which you can see here. I'm really enjoying every day working with all of them together. I would like to thank all of our collaborators on all of the projects. And I would like to, again, like to thank you for your attention, and I'm happy to take any questions. Thank you very much.
All right. Well, thank you so much, Carol and Christoph, for your informative presentations. I also want to welcome Joe Maffei here, as we will now be starting the live Q&A portion of the webinar. So if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. We've already got some great questions coming in here from the audience. So let's start with this question here. This question asks, thank you for the informative talk. I was wondering if there are any difficulties with detecting certain classes of targets, or is each gene equally detectable?
Yes, I don't know. Do you want to answer this?
Sure.
Yeah,
I'm happy to answer it. So just as a quick introduction, my name is Joe Maffei. I'm one of the field application scientists with Vizgen. So as as the talk profiled, you saw the probe binding approach that we take, and how we use probes to tile across RNA transcripts to detect different species of RNA in high plexity. That approach requires many different probes to tile across any given RNA transcript, and therefore we need a certain amount of real estate on every transcript in order to detect it. So when we talk about certain classes of targets, one class of target that might have detection efficiencies that are reduced would be small RNA transcripts. So we're talking about really short genes where we would not be able to tile across the full 50 probes per transcript that we like to detect. And along with that, we do need to make sure that between every gene that we're detecting, there is enough unique sequence across different transcripts so that we can distinguish between them. So as long as those conditions are met, all of the genes can be detected with high efficiency. But just like with any other technology, there are limitations and boundaries that we come up against based on the way that the technology works. But all of those targets that may be quote unquote more difficult, those are things that you can always have a conversation with your application scientists, the bioinformatics folks at Vizgen, to gain a better understanding as to how to build your panel to make it in a way that's going to be best for your project.
Great, thank you, Joe. All right. Next question here asks, what is the typical section thickness recommended, and how does the pre-processing pipeline take care about, oops, slipped up here, one moment, sorry about that. I'll repeat the question. What is the typical section thickness recommended, and how does the pre-processing pipeline take care about potential overlaid cells?
Yeah, so the thickness that we recommend is different depending on whether you're working with a fresh frozen or frozen tissue or an FFPE. So for frozen tissues, 10 micron thickness is our typical thickness that we work with, although the system does allow for some wiggle room there because we can change the imaging depth based on how thick your sample is. For FFPE samples, the slices from those blocks are usually much thinner. So we run with about 5 or 4 micron is our typical thickness for that. So in terms of the pre-processing and filtering of cells, we have our algorithm for performing the cell segmentation is fairly sophisticated, and it uses lots of different approaches to marry cell boundaries from different z-planes. So for every experiment, we do take multiple z-planes, and each one of those z-planes is treated as its own separate image. We can draw boundaries around cells in those z-planes and then marry each one of those boundaries back together to create a cell that spans over a small 3-dimensional space. So all of that is worked out by the analysis computer during the segmentation phase. So that's not something that's requires user input. But all of the boundaries that are drawn are available to you as a vector format. So you can look at them, you can plot them in the visualizer and see if it looks good to you. And if you feel that there is some manipulation necessary for the segmentation, Vizgen has a post-processing tool that works on the command line that allows you to customize the segmentation options. So we're talking about changing cell pose parameters and then using different stains channels to create the best possible boundary for the segmentation.
Great. All right, moving on here. This next question asks, can this technique distinguish splicing variants?
Yeah, so I can take that one too. So this kind of goes back to one of the previous questions that was asked. So I'm very glad for the opportunity to talk about that. So I mentioned earlier that when we build probes against any RNA transcript, we tend to build about 50 probes that tile across the length of the RNA transcript. And the uniqueness of the probe binding allows us to distinguish between two different RNA molecules. So to answer your question kind of bluntly, it really just depends. It depends on what's the variance between those two RNA transcripts. If it's something like a point mutation, there's probably not enough unique difference between those two RNA transcripts to distinguish them. But if we have a larger sequence difference between the two transcripts, whereby we can build a unique set of probes against each transcript variant, then that becomes much more of a possibility. And again, speaking and consulting with our bioinformatics experts is the path forward to generating a panel with those transcript variants.
Great, thank you. All right, this next question asks, for a genetically modified kidney tissue or diseased tissue, would you recommend performing scRNA or snRNA-seq prior to running MERFISH to gain a better understanding of which targets to specifically use for better visual differences and characteristics between a normal kidney?
Yeah, I guess I'll take that question. I mean, that always depends also a little bit on what type of material you have to start. So if the tissue is frozen, it's really difficult from, for example, a kidney biopsy to obtain enough whole single cells instead of nuclei. That's sort of a limiting factor. Instead of, and if the question is there an information difference that is needed to map out cell states between these two single cell or nuclei approaches? I would say no. If you have the option of going for a full-length information assay, I would say yes, because the full-length RNA transcript single-cell information assay can actually also give you information about, for example, the 3' UTR length and other things that's usually not, depending on which enrichment method you're using for the nuclei, is not so, these data then might not have a nice coverage. And they can be informative to, pick genes or targets that are more likely to be spatially differently localized within the cells. So in short, in two sentences, single nuc RNA-seq to map out single cells would work equally well compared to whole cells. But it depends on your further downstream question of what you want to use that data for panel design. If it's about special things about spatially distributions of transcripts within your target tissue, then I would guess it's more about which full-length 5' or 3' assay you are using.
Thank you, Christoph. All right, this next question asks, for the panel design you mentioned SPAPROS from Theus Lab, what is the gene panel, or what if the gene panel includes too many high expressed genes? How do you detect this?
Yes, so that happens or can happen quite frequently, right? I mean, the good thing is if you have a reference data set, you find usually co-regulated genes, right? So some of the genes are, you can basically use SPAPROS to swap out genes. It's actually implemented in the pipeline quite efficiently to select as informative genes for cell state, but they are lower expressed, and that works actually quite well. So that way you avoid to pick too many high expressors to avoid optical crowding. So this is actually implemented within the pipeline of SPAPROS. And this also takes into account the other things that Joe was mentioning. So does the transcript have the optimal length to be able to target enough probes in there?
I can also add to that as well. And to that was a very good answer. So one of the things that Vizgen can do with their panel builds is stick some of these really expressed genes into what we call a sequential panel. So what that does is it images these highly expressed genes individually from the rest of the MERFISH panel, so they don't interfere with the optical crowding. The output for those genes is slightly different from the MERFISH genes, but you can use them for things like labeling cells and identifying cell types. So if you have highly expressed marker genes that you want to use, it's not it's not something that you have to necessarily avoid totally.
Great. All right. Next question here. What would be the procedure to design custom panels of genes, and how much flexibility do we get to select the genes of interest?
Sure. Yeah. So building a gene panel is, you can make it completely custom, which is one of the strengths of our company right now. Up to 500 genes, you get to select every single gene that belongs in your panel. And most of the time that's done through our portal. So our portal is this online cloud software where you create an account, you log in, and the gene panel process is as simple as just telling us the names of the genes that you want in your panel and specify your type of interest. So that goes through a bioinformatic review, and the entire finalized panel gets sent to our processing facility where we would start to build it custom. So you do have the option of building everything out from scratch in a way that fits your project best.
Awesome. Great. Well, thank you for that answer there. We have time for a few more questions here, so we'll continue. This next question asks, how did you validate the antibodies? Did you validate them individually or in a pool? And how was the overall success rate?
Yes, so I mean, for the we tested different antibodies on FFP tissue up front to the MERFISH experiments. So, I mean, any antibody that, so in our experiment, any antibody that works for standard immunofluorescence on FFPE tissue is also very likely to work also with the MERFISH or with the Vizgen's pipeline for protein staining using the secondary antibodies that I've shown and which are sold by Vizgen. And obviously, of course, the evaluation upfront using just FFPE tissue is something that most researchers are very familiar with. But of course, you would want to know before you run the actual experiment if the antibody staining is going to be successful or not. And there's a particular antibody staining or protein staining kit that you can use just for this purpose that allows you to test if your primary antibody actually really works. And you can obviously, depending on the species of the antibody, multiplex several of those primary antibodies together in one run to see already if, for example, a double or triple immunofluorescence will turn out successful, which we found very useful because that's what most researchers like to do is, of course, not measure only one particular protein target, but actually have several ones. And so you can evaluate that directly in one run.
Great, thank you. All right, next question here. You mentioned multiplexed protein staining using Vizgen's protein staining kits. So how does this exactly work?
Yes, so what you do, it's actually very easy. So the antibodies, I mean, you have to have your own antibody, your primary antibody. And then before you start the actual MERFISH experiment, you stain your tissue before the gel embedding with your primary antibody and the secondary antibody. And then afterwards, so it's like a standard procedure, just as you would for normal immunofluorescence staining. It's not anything particularly difficult, definitely not. And then once you finished the washing steps after the secondary antibody staining, you can just proceed with the normal protocol. So in that sense, it adds this antibody staining parts to the protocol, but they are, I would say, very routine to any sort of experienced scientist who has performed immunofluorescence stainings before. So.
Awesome, great. Well, thank you, Christoph. Looks like we have time for one more question here, so we'll wrap up after this one. This final question asks, regarding the data analysis and exploration, is this done exclusively on a cloud-based software, and is the access free and unlimited?
Yeah, maybe I answer this. So this is entirely open, right? I mean, you get your raw data and also the process data from the instrument. So what is after what you do is a processing where you select basically the optimal cell boundary. If you've included it, you can include DAPI or different cell boundary options for the segmentation. And then this segmentation is actually run locally on a workstation, a powerful workstation that processes very fast. So you, as a researcher, get the even segmented and sort of decoded, not the raw data, but really the data that you will use for downstream analysis quite fast after the run. This doesn't involve any cloud service or any other things. For this reason, you wouldn't even have to hook up the machine to any network. Of course, for data transfer, this would be recommended then later. But you could also do that with a hard drive and the hard drive connection on the machine, which is also quite fast.
Great. All right. Well, this will conclude our session for today. So Christoph, Joe, do you have any final comments for our audience?
No. I think.
I guess I would just say thanks to everyone who joined. And if you do have other questions about Vizgen or about the MERScope, definitely feel free to reach out to us as a company because we're interested in talking with you.
Great. Well, thank you again, Carol, Christoph, and Joe for your time today and your important research. We would also like to thank Labroots and our sponsor, Vizgen, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.